All right, in Chapter 17, Part 3, we talked about the translation. So in the AHL portion of Chapter 17 for Part 4, we're going to talk about the idea of translation and how the whole kind of process occurs. So translation, of course, is when the cell is going to take that message from the messenger RNA and interpret it into a protein. And the kind of the mediator, the go-between, the kind of the actual interpreter is the transfer RNA. That's going to read the messenger RNA with the help of the ribosome and bring in the correct amino acid from the cytoplasm. Okay, and all transfer RNA molecules are different. So when we look at this process, this is occurring in a bacterial cell, of course. This process of transcription and translation, when we look at this translation part, it's occurring right here, of course, at the ribosome. Okay, and that's where we're going to be kind of focusing what we're talking about here with what's going on. So the transfer RNA is made in the nucleus. It's used over and over again. So it brings in the amino acid, it forms the peptide bond, and it kicked out of the E site. And on one end, you can see this is where the amino acid is actually going to attach. And then on the other end, this is the anticodon. This is the part where it's going to actually read, say, the messenger RNA molecule to ensure that we've got the correct amino acid actually being brought in to the ribosome. And when this transcript is read, this is when the amino acids begin to kind of do their part in forming the polypeptide. They might be interacting together, giving it some secondary or tertiary shape. And the two important steps really in the recognition of this translation is that there has to be a correct match between the transfer RNA molecule and the amino acid, and then there has to be accurate translation at the messenger RNA molecule. Okay, so with the correct match, the first part here, we're going to use a little bit of this ATP energy. And what we're going to do is we're going to join up an amino acid to the transfer RNA molecule with the help of this aminoacyl tRNA synthase molecule. Okay, and there's 20 of these, one for each amino acid, and the enzyme is going to catalyze the formation of that with the help of a little bit of energy. And now this amino acid here is activated on a transfer RNA molecule and ready to be taken to the ribosome, where they're brought in. And of course, there's not a one-to-one ratio of transfer RNA molecule with the messenger RNA. So there's, the redundancy that we were talking about, or the wobble, because there can be kind of some forgiveness. But once these things here are brought in with the anticodon, we're going to look at the codon to make sure that there's a correct match. And if there is, then we're going to kind of shuffle it along into this direction, which is where this peptide bond is going to form. And then we're going to shuffle it along to the E, where it's going to exit, and then go back and kind of repeat this process that we've been talking about. Okay, so that's the accurate translation. And when we refer to, for instance, with wobble, we can get some forgiveness in one of the base pairs. So this is why, when you look at the codons for some of the amino acids in their third base, like for instance, if we look here, the third base of leucine can accept UUA or UUG. So it can accept an A or a G in that third place on the messenger RNA codon, and that's called wobble, and that's some of the forgiveness that's actually occurring. Okay, so if we look at ribosomes now, this of course is where there's protein synthesis, and it's important to know that there's two different kinds. There's a kind that's found in the bacterial cell. There's kinds that are found in the eukaryotic cell. And the bacterial now consist of 70S ribosomes. They have a 50S and a 30S subunit. And then they have an 80S, which are found in the eukaryotic cells, and they consist of a 60S and a 40S. Okay, so these are the two different types. And of course, if you remember, there's a large and a small subunit on these ribosomes. Okay, so if this were a bacterial cell, let's say, it would be 50S. And this one here would be 30S. Okay, and then together I know they look like 80, but they equal 70S. Okay, so that's the 70S subunit. The S is just a Svedberg, it's just a unit. Okay, and then the eukaryote, of course, the large would be 60, the small would be 40, and that would give an 80S. Okay, all right, so that takes care of the ribosome and kind of explains a little bit about, really some cutting edge research, what people are doing, and at least we're doing, 4 or 5 years ago, but they're still working on now. Synthetic life form has been made. George Church, I believe, was the second person to do it. Craig Ventnor and his team in the West Coast were able to do it first. But just a little bit more importantly, a little bit about the ribosome. You can see how it works and, some of the research that's involved in doing that sort of a thing. So when you talk about ribosomes now, there's two types, if you recall, from when we learned about this back in Chapter 6. There's free and bound, and they function the same. They're making proteins. And the switch from free to bound, can happen, and really it's what it has to do with is the protein that's being made and whether or not it contains a signal particle that's going to instruct it to go to the ER, or whether it doesn't contain that. And if it doesn't, then it's just going to make proteins that are freely available to use by the cell. Okay, and of course you recall once it's attached to that ER, it's going to make proteins that are going to be exported from the cell. So if you look here, here's the ribosome, of course, and then what I'm going to do is just switch colors here. Here's the ribosome, and this is going to read this messenger RNA molecule, and when it does, it's going to start to synthesize this protein, and on the very end of it, it's going to have this signal peptide, and that signal that peptide is going to bind with the signal recognition particle, and that's what's going to be responsible for basically hauling this entire kind of complex, the messenger RNA, the ribosome, as well as the growing peptide, hauling that to the ER. And now what this is going to do is it's going to continue to make the protein, and in doing so it's going to push it into the lumen, or the interior of the ER. And now once this whole thing is done, it's contained within the ER, and now it can get shipped off to the Golgi for processing, packaged in vesicles, and then of course shipped out of the cell. Okay, so again, just a little bit about that signal recognition particle. This whole complex, like we said, is going to bring that ribosome to the ER so that it can be made in the ER, and then ferried along and gotten rid of out of the cell through a vesicle. Okay, so this is just a couple of things that are just showing that process again. And once the entire protein has been made, there's an enzyme that's going to be involved in cutting off that signal recognition particle, so that you're just left with an actual protein that has what it needs, and it can go to the Golgi and be instructed on where to actually go. Okay, so again, just a couple of things to reiterate here. Some of the differences between the prokaryotic and eukaryotic gene expression is that the RNA polymerases, of course, are different, but they function effectively the same. Transcription is terminated a little differently between the two. We talked about that, I think, in part 2. Prokaryotic and eukaryotic ribosomes are different in terms of their size, not in terms of what they do. The ribosomes in the bacterial cells are a little smaller. The ones in the eukaryotes are a little bit larger, and that enables some antibiotics to work effectively in prokaryotic cells by targeting those ribosomes because they're different, yet not have any effect on eukaryotic ribosomes, which is good for eukaryotes that get infections in treating them with antibiotics. And then transcription and translation in a prokaryotic cell streamline. They can occur at the same time because there's no nucleus. In a eukaryotic cell, they occur, of course, at different times. And then eukaryotic cells, of course, have a complex system of kind of targeting proteins for their final destination. Okay, so one of the final notes here, just remember that not all DNA codes for proteins. Some of this regions of DNA are involved in other aspects of metabolism, kind of regulating things, turning on and off genes, etc. And certain portions of that DNA, again, code for transfer RNA molecules, of course. The ends of DNA, of course, are called telomeres. Some of the DNA is intronic. Some of it is, comes from viruses, and it's just been incorporated through over the millennia, etc. So not all DNA codes for protein. It's important to remember those types of things, and it's also remember a couple of examples of DNA that doesn't code for protein, such as the stuff that I've given here. Okay, so that wraps up all of the AHL material for Chapter 17. Be sure, of course, to write down any questions that you have, and we'll definitely go over them in class. Okay, so we'll talk to you in a little bit.